Hey everybody, Dr. Viso here. In this video, we're going to talk about T cells. And so this is a pretty big video. We'll talk in the beginning about T cell development. What are the big things you have to know in regards to, how T cells develop, positive selection, negative selection. These tend to be particularly high yield. We'll talk a little bit about the MHC or major histocompatibility complex 1 and 2, and then we'll talk about the process of T cell activation. If you've been following this video series so far, again, everything is really going to start here in the bone marrow, and we're going to have, CD34 positive hematopoietic stem cells that eventually form the myeloid and the lymphoid lineage. And we're primarily concerned, especially in immunology, with the lymphoid lineage. And this lymphoid lineage is going to give rise to T cells, to B cells, to natural killer cells. Now, the B cells, remember, the B cells are going to not only originate in the bone marrow, but they'll also then go on to kind of develop in the bone marrow. But the T cells are a little bit different. They'll also originate in the bone marrow because they all come from the lymphoid progenitor cells, right, which are going to be found in the bone marrow. However, the T cells are a little bit different in that they'll develop in an outside lymphoid organ, and that is going to be the thymus. And you might be saying, well, how do you even get these lymphoid progenitor cells over to the thymus? Well, there's certain chemokines that are released that essentially are going to signal the lymphoid progenitor cells to shuffle over some of their inventory over to the thymus so that we can develop some T cells. Okay, so this lymphoid progenitor cell is going to leave the bone marrow through the bloodstream and eventually go to the thymus. Okay, so that's what we have here. It leaves the bone marrow, goes to the thymus. And at this point, this cell is CD4 negative, CD8 negative. There's no CD4, there's no CD8 on this cell. And these are two markers you want to be really familiar with a T cell, particularly in the stages of development, because different stages, there'll be different receptors, and this tends to be confusing. And the way I remember it is, look, this is a lymphoid progenitor cell. We haven't started kind of classifying it as a T cell until it gets to the thymus, right? And so at this point, we would expect it, as it arrives to the thymus, to be CD4 negative, CD8 negative. Now, once it gets to the thymus, though, there's going to be certain enzymes, certain genes that are going to stimulate shuffling of some of the genes in this precursor T cell. And the one that you really want to be familiar with is going to be RAG, which is essentially this recombinase activating gene. If you think about, a recombinase enzyme, what are we talking about? We're talking about taking the DNA, shuffling it up, mixing it up so that we get some variation and some diversity in those genes. And the idea here is, if we're shuffling up all of the DNA here in this precursor T cell, we can create a very unique T cell receptor that will bind to a unique antigen. If we create a whole bunch of different T cell receptors, we're going to have more immunity to different types of antigens. And that's essentially what these RAG genes are doing. And so the two big recombinase genes, like we said, is going to be RAG1 and RAG2 that are going to be kind of stimulating this process, this recombinase process, to eventually lead to the development of the T cell receptor. Now, remember what I said, you do have to keep in mind where's CD8, where's CD4. And you can see here that this precursor T cell, after it comes into the thymus, it's going to have that recombinase gene causing the formation of a T cell receptor. And with that T cell receptor formation, there's also going to be the addition of CD4 and CD8. CD4 and CD8 are essentially co-receptors to the T cell receptor. Okay, so we're basically putting everything on this precursor T cell, including CD3. CD3 is usually going to be, in terms of board questions, a marker to tell you that you're dealing with a T cell. It doesn't necessarily tell you what type of T cell like CD4 and CD8 will, but CD3 is kind of just like a general T cell marker. So you're going to get a T cell receptor, CD4, CD8, and CD3 all placed on this precursor T cell once it gets to the thymus. It's essentially the body's way of saying, look, now that we've got this lymphoid progenitor cell here, we're marking this as a possible T cell. Okay, and so that's what all of this stuff going on to this T cell is for. Now, as you'll see in a minute, we'll eventually have a CD4 receptor or a CD8 receptor along with the T cell receptor and the CD3, but we won't have both CD4 and CD8 when this process is done. But in the beginning of the process, we have both of these receptors. And so sometimes these precursor cells, once they get to the thymus and they have RAG1 and RAG2 activation, they'll be called double positive T cells. Okay, and so just to recap this here, we have this initial pre-selection process, which I'll talk about in a minute. The T cell receptor will be created using RAG1 and RAG2, like we said, and it will be CD4 positive, CD8 positive. Now, the next step in this process is particularly high yield. This is positive selection. In positive selection, we're essentially trying to find the right antigen. You can think of it in some ways as a positive match in terms of an antigen that we can bind to that T cell receptor that we created using the RAG genes, RAG1 and RAG2. And so just to take this for example, what will happen is, again, the blue structure here is going to be your T cell receptor. And you can see that T cell receptor can bind to some of these thymic epithelial cells. Okay, so this would be a thymic epithelial cell. And these thymic epithelial cells will either have MHC1 or MHC2. Okay, now remember, the CD4 positive co-receptor will help that T cell receptor bind to MHC2. Conversely, the CD8 positive co-receptor will help that T cell receptor bind to MHC1. Okay, and again, this goes back to your rule of 8s. Okay, so 8 times 1 is 8, right? So we're talking about CD8 positive times MHC1. That gets you to 8. Now on the other side of that, 4 times 2 also equals 8, right? So CD4 positive cells that are going to bind to MHC2 receptors will also get you to 8. Okay, so that's the way to remember. CD8 positive cells are going to bind MHC1, CD4 positive cells will bind MHC2. And you might be saying, okay, hold on a second there, Dr. Visor. You said that this cell has CD8 and CD4. So how do I know which one is going to bind? In reality, one of these receptors will find their specific antigen on these thymic epithelial cells and they'll bind. And when they do that, they're going to essentially select themselves to either be a CD4 or CD8 positive T cell. Okay, so it's kind of, in some ways, it's a bit random how this happens. But the point is, the big point that you want to remember here is that in the process of positive selection, what we're essentially doing is we're finding the right fit. We're finding the antigen that binds specifically to this receptor. Now, if that antigen binds too tightly or it doesn't bind strong enough, what will happen is these T cells will eventually get an apoptosis signal. So if the binding is too strong or too weak, that T cell will eventually undergo apoptosis. You have to have just the right amount of binding to get to the next step, which would be negative selection. Okay, so in positive selection, you're looking for adequate binding. And again, the CD8 positive co-receptor will bind with MHC1, the CD4 positive co-receptor will bind with MHC2. So it's kind of completely random as to if, this precursor T cell binds to CD4 positive MHC2 or uses CD8 positive to bind to MHC1. But once that process happens, then we're essentially selecting the route that this cell will go down, okay? Once it chooses to bind to whatever specific antigen is present. And again, if the binding is too strong or too weak, then it's going to undergo apoptosis. But if the binding is just right, then we can get to the next step. And the next step is going to be negative selection. Now, negative selection, unlike positive selection, is going to take place in the thymic medulla. Again, we're going deeper into the thymus as we proceed here. Okay, so negative selection is the process of essentially, again, binding to the same type of receptors that we bound to in the initial step. But the difference in negative selection is that we're using our own self-antigen. So these medullary thymic cells, instead of presenting a foreign antigen, we're presenting antigens from our own body. And the idea in negative selection is we don't want our T cells to attack our body, right? We don't want our T cells to attack our own cells. And so we're presenting our own cells, kind of like a test to say, hey, we just want to make sure that you're not going to be attacking us. Okay, and so that's what this second step is. Now, if the T cells bind really tightly to our own antigens, that's concerning because then we're thinking, well, it's possible that these T cells could attack our own cells. Okay, so self-antigens, again, self-antigens being the key point, right? Antigens from our own cells presented on the MHC1 and MHC2 receptors. If strong binding occurs to self-peptides, again, our own peptides, then those T cells will likely undergo apoptosis. So you can see that again here. So if the binding is really strong, again, these T cells will then undergo apoptosis, okay? Now, the T cells that do not bind, right? So the T cells that do not bind, those cells will then go on to form mature cells, okay? So again, this being the example of the CD4 positive T cell, and then this being the example of the CD8 positive T cell. And again, in both situations, if there's no binding, then we go on to essentially have a matured T cell, okay? Once we complete negative selection, we have a matured T cell. Now, keep in mind, this T cell is still naive. What does that mean? Naive means that this T cell has not yet run into its antigen that binds to its specific T cell receptor in the body yet. Okay, once this T cell runs into that antigen, that T cell can then become activated. Okay, typically when a macrophage or dendritic cell presents that antigen to the T cell and activates it, then that T cell would no longer be naive. But at this point, it's important to understand the terminology. The T cell is mature because it went through this whole process. It's been tested for positive selection. It binds to its antigen, not too strong, not too weak, and it won't attack our own cells, most likely because we didn't have binding to self-peptides. Okay, so that's the concept. So that's why it's mature. It's naive because it has not yet run into its antigen in the bloodstream. So just keep that in mind, or in any lymphoid organ in the body. What you'll notice here at the very end of this, you might be saying, what happened to those CD4, CD8 receptors? Okay, so you can see here, we had a CD4 positive cell in negative selection. It still had on this CD8 receptor. So basically, throughout this whole process, these cells were double positive T cells. Through positive selection, through negative selection, they were double positive. But what will happen is, after the selection process has occurred, you'll start to see downregulation of whatever receptor was not used during this process. So if we look on the left, this was mostly going to be the CD4 positive T cell because it bound to MHC2 initially, right? Going back to our rule of eights. So what happened is eventually you'll see that this CD8 coreceptor eventually falls off as it matures into a mature T cell. Okay, so when you get to a mature T cell, you don't have CD4 positive, CD8 positive. You have one or the other. It just depends on which pathway this T cell went down. So you can see here, once you have a naive T cell post-selection, an immunocompetent mature T cell, it's either going to be CD4 positive, CD8 negative, or CD4 negative, CD8 positive. Again, if it's CD8 positive, we call those cytotoxic T cells, CD4 positive T helper cells, and we'll go into a lot of detail on this in the next couple videos. But the point is that there's downregulation of that receptor after we go through selection. And the last thing that I want to touch on here, remember we talked a little bit about these Tregs or T regulatory cells. Now, a small percentage of these cells that actually do bind to self-antigens will actually stick around. So you might be saying, what are we talking about here? We're going back to this step at negative selection. Okay, so right here, again, we presented our own antigens, a self-antigen. We don't want the T cell receptor to bind, right? When the T cell receptor binds to that, we basically are saying, hey, this thing has got to go. It's got to undergo apoptosis because it could attack us. A small percentage that actually do bind don't undergo apoptosis and instead go on to form regulatory T cells. Okay, and this is particularly classic for the CD4 positive line here on the left. So what these T regulatory cells essentially do is they help kind of damper down the immune response. They regulate the immune response, basically, so it just doesn't get out of control. Okay, that's kind of a simple way of explaining it. And again, we'll go into more detail on this coming up. But the important thing to know here when we're talking about this self-antigen presentation with regulatory T cells and negative selection is there's a very classic protein here, and that's going to be this ARI protein. And this is very classic because this particular protein is responsible for allowing this process to happen. In other words, it allows us to give self-antigen presentation so that we can kind of show these T cells, hey, this is what our antigens look like. If this protein is not functioning accordingly, if there's a defect in this, then you can imagine we might have a higher risk of essentially attacking our own cells because we're not able to effectively undergo negative selection. And the classic pathology that this manifests as is going to be autoimmune polyendocrine syndrome, which is essentially a combination of a bunch of different endocrine-like disorders due to this autoimmune response where we're attacking a lot of our own cells, leading to endocrine dysfunction. So you get things like adrenal insufficiency, you can have hypoparathyroidism, chronic mucocandidiasis is also very classic. And again, we'll probably talk about that in greater detail, but just kind of keep that in mind, big picture. Okay, so just to recap this, right, we had some lymphoid progenitor cells, we had chemokines that brought that lymphoid progenitor cell from the bone marrow where it originated to the thymus to mature. Once it got to the thymus, CD4 negative, CD8 negative, the recombinase gene shuffled up the DNA, we made a T cell receptor, and it was double positive at that point. And again, I want to say that all of these, if you look at all of these, they all have this CD3 receptor on them. So throughout this entire process, right, we also had a CD3 receptor, and that stays on all of these cells even when they're mature. Remember, CD3 is very classic for marking a cell as a T cell in general. Okay, so remember that. But the CD4 and the CD8 receptors, we're going to basically go down one of the two paths where we end up keeping one or the other. Okay, so again, if we initially are going to bind to MHC2, thymic cortical epithelial cells will then probably go down the CD4 positive route. If we bind to MHC1, we'll go down the CD8 positive route. You can always remember that with your rule of eights. Okay, so the first step was positive selection. We wanted to see if there's adequate binding to an antigen that is not self, right? Some other foreign antigen usually. And if there was adequate binding, the T cell will go to the next step. If not, it would undergo apoptosis. With adequate binding, the T cell moves to the medulla. Then there's negative selection. We present self antigens largely driven by this protein, the Aerie protein. And if there's binding to self antigens, then that cell needs to undergo apoptosis because we don't want it attacking self. Some of the small percentage of those cells will become Treg cells to help regulate the immune response. And then eventually, once this process is completed and this cell survives, then it will become a mature T cell, either CD8 positive, CD4 positive, and then downregulate the other core receptor. It will still have a T cell receptor and it will still be CD3 positive.